Good day, Honors Biology students. Welcome to video lecture episode 16. Remember, we're now in chapter 12, and in particular, section 1 of chapter 12 called DNA, the genetic material. Remember, last time we had just barely gotten into the first discovery that was working on discovering what the genetic material was, and that was Griffith's study of pneumonia. Well, what Griffith deduced from his study is there must have been a transformation from the live R strain bacteria to the live S strain bacteria. Okay, remember R was the rough and the S was the smooth. If you need to be reminded of what they look like, you can go back to slide 16 and you can see the image that I have there. Well, what Griffith deduced from his study at this point is basically there must have been a transformation. In other words, it changed from one to the other. This he noted as the disease-causing factor was passed from the killed S bacteria to the live R bacteria. All right? So let's talk a little bit more about what his experiment means. And it's this. This experiment that Griffith did set the stage for the next development, that being discovering the identity of the transforming substance. In other words, he laid the groundwork and began the path in the right direction, so to speak. Next up in the path to the discovery of the genetic material of inheritance is a man by the name of Oswald Avery, and he's there on slide 22 on the right-hand side of that image there. Avery and his colleagues in 1944 discovered the molecule which transformed the R strain into the S strain. All right? Now, Avery in his experiments isolated different macromolecules such as DNA, lipids, and protein from the killed S cells. He then exposed each of those macromolecules individually to the live R cells. All right? Now, when he exposed the live R cells to the S strain DNA, it caused a transformation. The R cells became the S cells. Despite this experiment and despite what it seems to be conclusive, Avery's conclusions were not widely accepted within the scientific community. So, let's move forward in history. In fact, let's fast forward 8 years. We come to the year 1952. Two scientists by the name of Alfred Hershey and Martha Chase. Hershey and Chase published results of experiments they had conducted in connection with searching for the genetic material. What they provided in their publication was definitive evidence that DNA is the transforming factor. Their experiments involved a bacteriophage. This is a type of virus that attacks bacteria. Now, if you look on slide 27, I have a picture of it there, as well as slide 28 and 29, which are some a little more updated pictures, computer graphic-ish. All right? Now let's move to slide 30. In their experiment with bacteriophage, there were two main components which made the experiment ideal for confirming DNA as the genetic material. All right? One, the virus they were using, bacteriophage, was made with both proteins and DNA. And two, viruses cannot replicate themselves. All right? Viruses, in fact, must inject themselves into a living cell to replicate themselves. Hershey and Chase therefore labeled both parts of the virus, the DNA and the protein, to see which part was injected into the bacteria and thereby show what the genetic material was. All right, ladies and gentlemen, we're going to go ahead and conclude our lecture here. If you have any questions or comments, you can leave them below. Reach out to me, send me an email. I do hope it's been helpful. Have a nice day. Take care.